Okay, so chapter 14, we're going to look at the structure of DNA, which is one of the nucleic acids. DNA stands for deoxyribonucleic acid. And we're going to look at the structure of DNA and the structure of nucleotides, which are the monomers of DNA. And then we're also going to look at how DNA is replicated. So you will remember that prior to any cell division, the DNA has to be replicated so that each of the product cells can get the copies of the chromosomes. And so it's going to be relevant for binary fission in bacteria. It's relevant for eukaryotic cells. So how the monad chromosomes become dyad chromosomes. That's what DNA replication is about. So first we're going to look at the structure of DNA and the structure of its monomers, which are called nucleotides. So DNA is a nucleic acid, a polymer made up of monomers called nucleotides. Each nucleotide has a 5-carbon sugar called deoxyribose, a phosphate group, which I hope you still remember what that is, and then a nitrogenous base, which is an organic compound that contains nitrogen in it, although that's not that specific. And DNA has one free hydroxyl group at the 3' position of the ribose sugar. And I'm going to show you on the next slide that RNA nucleotides have 2 hydroxyl groups. So in this slide, I'm showing you really the nucleotide details of both DNA nucleotides and RNA nucleotides. Although we won't talk more about RNA nucleotides until the next chapter. So fundamentally, after this, we'll be talking about the DNA nucleotides. Okay, so you can see here, this on the left side, you have an example of a nucleotide. There's always a phosphate group here. You remember your P surrounded by oxygens all the way around. That's your phosphate functional group. Then you have, that's attached to what we call the number 5 carbon of ribose, which is shown here as this little pentagon. So just like glucose has several carbons in the ring and then one that sticks up out of the ring, this sugar, which is ribose, it only has 5 carbons altogether, but 4 of them are in the ring and then 1 of them sticks up out of the ring. So it's a monosaccharide, but this monosaccharide is attached to phosphate here. And it's also attached to something called the nitrogenous base here. So we say the number 1 carbon, which is where my pointer is, the number 1 carbon on the ribose sugar is attached to the nitrogenous base. The number 5 carbon on the ribose sugar is attached to the phosphate. Now, if this is a DNA nucleotide, then this little green box here would just be a hydrogen. You could just write H in this spot. So in a DNA nucleotide, the number 2 carbon is attached to a hydrogen. In an RNA nucleotide, the number 2 carbon is attached to a hydroxyl group, OH. That doesn't seem like that much of a difference, but in fact, it is very important for the stability of the polynucleotide strand. However, for us right now, all you have to know is that what the difference is. There's a hydrogen here in DNA nucleotides. There's a hydroxyl here in the RNA nucleotides. So you should write that down. They both have a hydroxyl group on the number 3 carbon. So attached to the number 3 carbon, both RNA and DNA nucleotides have a hydroxyl group. So this hydroxyl group here on the number 3 carbon is very important. And this phosphate group is very important. So we'll be talking about those in a minute. Let's focus on this nitrogenous base for a second. You have really 2 types of nitrogenous bases, ones that are called the purines and ones that are called the pyrimidines. The purines are adenine and guanine, and both adenine and guanine have this ring structure. You see how there's carbon and nitrogen and hydrogen. And these nitrogenous bases have a 5-membered ring attached directly to a 6-membered ring. So sort of a little pentagon attached to a hexagon. Same thing for guanine, a pentagon and a hexagon. What's different about these two, the guanine and the adenine, is what's attached on the outside here. So this one has a C double bond O in guanine, but adenine has a C with an amino group attached. And there's some others like that, some different functional groups attached. And that does make them slightly different chemically. But you don't have to know the detailed structure, just the general fused ring structure of the purines. Now the pyrimidines, they have a simpler structure. They just have a single 6-membered ring. And again, though, attached to some of the members of the ring are different functional groups. So this one has a C double bond O. This one also has a C double bond O, but this one has a C bound to an amino group. And there's other, you can see some differences around if you look. So there's some differences, and these three are called pyrimidines. Cytosine is one of them, thymine is one of them, and uracil is one of them. And so you need to know the general structure, sort of the skeletal structure of the purines versus the skeletal structure of the pyrimidines. And then you need to know that adenine and guanine belong in the category called purines, and cytosine, thymine, and uracil belong to the category called pyrimidines. Okay, so let's talk about how two nucleotides would be bonded together. It's going to be a dehydration synthesis reaction that attaches these two together. The enzyme that attaches two nucleotides together is called DNA polymerase or RNA polymerase. So let me write that one down. And it just creates, it's this the molecule or the enzyme that creates a polymer. So DNA polymerase, Polymerase, that would be the enzyme that creates from individual nucleotides. It joins them together in a dehydration synthesis reaction and it creates a polymer. So DNA polymerase. And the same for RNA. RNA polymerase is the enzyme that builds a strand of RNA from individual nucleotides of RNA. And the connection then between the two, I told you was going to be important that you pay attention to. Remember, this is the number 1 carbon, this is the number 2 carbon, this is the number 3, this is the number 4, and this is the number 5. So every nucleotide has that same numbering, 1, 2, 3, 4, 5. So in this nucleotide, you have a nitrogenous base, and they're showing a pyrimidine. You have the deoxyribose sugar, and then you have this phosphate group here. And then in this nucleotide, you have a nitrogenous base, a deoxyribose sugar, and they showing the phosphate group here in a smaller form. But what happens is the hydroxyl group that was here on the number 3 carbon, this was a hydroxyl group, interacts with the phosphate group on the number 5 carbon of the adjacent nucleotide. So before they're connected together, they come close to each other. The 3' hydroxyl on this nucleotide and the 5' phosphate on this nucleotide, and that's where the dehydration synthesis is going to happen. And a bond is made and water is formed. So you'll remember a little bit about dehydration synthesis. And then when that all happens, then these are connected. So when that reaction is completed, then these two nucleotides are now joined together. And what you can see is there's another hydroxyl group here. And so if I had another nucleotide that has a phosphate group here, I'm just going to write P, and then attached to another sugar and another nitrogenous base, so pretend like this is another nucleotide, then this phosphate group will interact with this hydroxyl group and you can get another one attached. and then another one will come underneath and get attached to that one, and so on and so forth. So in a very quick period of time, you can have a bunch of these being added, and you can create this long chain of nucleotides. So DNA polymerase would be the enzyme that would do this. And if you have a long chain of nucleotides, then it's called a polynucleotide chain. Now, no matter how long the chain is, at one end, at one far end of the chain, you're going to have a phosphate group that's not attached to anything else because it's at the end of the chain. And we call that the free 5' phosphate. So in this drawing, it's oriented up here at the top. And no matter how long this chain is, when you go to the other end of the chain, there's going to be, I'm trying to draw a hydroxyl group. Now I'm just going to put an arrow here. This is the free, F-R-E-E, 3' hydroxyl. So we say that the strand of DNA has one end that's called the free 5' phosphate end, and the other end is called the free 3' hydroxyl end. And that's what we mean when we say that a chain of nucleotides has a 5' to 3' orientation. One end has to have the free 5' phosphate. The other end has to have a free 3' hydroxyl. So in right around 1950, scientists were trying to figure out the structure of DNA because right around 1944, it became clear that DNA is the genetic molecule, an important genetic molecule. And so scientists really started focusing on this and trying to figure out the structure of DNA. And not that much was known about it. And so one of the scientists named Chargaff did a series of experiments. He collected as much DNA as he could from all kinds of different specimens. And he broke all of those chains of DNA down into the individual nucleotides. And then he was able to separate the different nucleotides apart from each other and figure out how much of each nucleotide made up that original sample. And when he did this for lots and lots of different DNA from different specimens and different organisms, he found that there was a rule, kind of a standard pattern that he was seeing in his data, in that however much adenine he was finding in that particular specimen, there was always an equal amount of thymine. And these are those nitrogenous bases, you'll remember. And no matter how much cytosine he found, there was always about an equal amount of guanine. Now, we're not saying that it had to be equal amounts of all four. And when we use percentages, if we say, okay, 25% of the DNA contained adenine, 25% contained thymine, 25% was cytosine, and 25% was guanine. Yeah, that happened in one specimen. But in other specimens, he had other numbers. For example, one species might have 20% of the specimen contained adenine, 20% was thymine, 30% was guanine, and 30% was cytosine. So it doesn't matter what the numbers are, as long as the amount of A always equaled the amount of T. That was the pattern he was seeing. And then however much G there was also that equal amount of C. So that was a pattern he saw. And then maybe in a different species had 45% A and 45% T, but only 5% C and G. So what I'm saying is he's taking a long chain, a long amount of DNA, breaking it up into the individual nucleotides, and then putting the nucleotides into 4 containers. And out of the total, what percentage of the total was adenine, what percentage of the total was thymine, what percentage of the total was guanine, and what percentage of the total was cytosine. Of course, all the total has to add up to 100%. All right, so what he saw was a pattern, not in particular numbers, but in which nitrogenous bases were always equal to each other or balanced. And these became known as Chargaff's rules. Usually we write it kind of shorthand. We say %A equals %T and %C equals %G. And then all of 4 of these numbers have to add up to 100% because they're all a fraction of the total DNA from that specimen. So even with Chargaff's rules, we didn't quite know what to do with that information yet. It wouldn't be long, though, before scientists would be able to put things together. In the around the early 1950s also, we have a scientist, Rosalind Franklin, who was working in a lab studying the structure of DNA, and she was doing X-ray crystallography, which is a method that's still used today. You need a crystal, and actually DNA makes a very nice crystal. You need a crystal, and then you put it on sort of a little stand, and then you shoot X-rays through it, and the X-rays are scattered when they hit the crystal, and behind the crystal you have this X-ray film, and so then when you develop the X-ray film, you can see the scatter of the X-rays that the crystal of DNA caused. But in any case, she generated this very famous picture, and she was able to figure out some really key pieces of information about DNA. And she's credited with figuring out that DNA is helical and that DNA has a standard width. In other words, the diameter of DNA is uniform all the way down, which at the time they didn't know. Before that, they didn't know if that was the case. So she did some very key experiments. Some other scientists were working on the DNA question, and 2 of them are James Watson and Francis Crick. And they were actually not supposed to be working on the DNA question. They had other projects that their boss wanted them to work on, but they were very interested in the structure of DNA, and so they went around England. Actually, Watson was American, but he was working in England, and Crick was British, and they went around and talked to people and gathered information. They did not perform any experiments on their own, but they were able to put together a whole bunch of data, and then they would go back to their lab and build these models using materials in the laboratory, and they were trying to build these models to scale and see if the measurements made sense. But in any case, they eventually put together some of Chargaff's data, Franklin's data, and they proposed the structure of DNA in 1953. So they are credited with the figuring out the structure of DNA, and I'm going to show you what it is. First, I'm going to show you a single strand of DNA, but actually what Watson and Crick came up with is what we call the double helix. So any one strand of DNA consists of an unbranched polymer of nucleotides. So just like I showed you in that other slide, this would be one nucleotide here. You've got the phosphate group, you've got the ribose sugar, deoxyribose sugar, and you've got the nitrogenous base. This is another nucleotide. This is another one here. This is another one here, and so on. So they're just showing 4. No matter how long this chain is, at the very top, you will have the free 5' phosphate group, which is shown here. And no matter how long this chain is, if you go all the way to the other end, you'll have a free 3' hydroxyl group. So we say that one end, we usually call the end, we always call the end with the free 5' phosphate, we just shorten it, we call it the 5' end. 5 with a little mark, 5'. And the end that has the free 3' hydroxyl group, we call that the 3' end. And so we say that the strand goes, that one end is 5', we mark it, we write 5' at one end, and at the other end we write 3'. So one end has to have the free 5' phosphate, and the other end has to have the free 3' hydroxyl. And usually they draw an arrow starting at the 5, and they draw an arrow towards the 3. That's just the standard. And so you can see what that directionality is. But that's just a single polynucleotide strand, and I said that DNA is actually two strands that we call the double helix. Double because there are two strands, and the helix because it spirals. So the important part of this is that the strands, the two strands, are oriented in what we call anti-parallel directions. In other words, if I were to take this double helix and straighten it out, unwind the helix so it's just flat like a ladder. I'm just going to flatten it out, the two strands, untwist it. Then what I would have is on one strand, it would go 5' would be here maybe, and 3' would be down here, no matter how long that strand is. And on the other strand, this one would be 3 here and 5 here. So basically, one strand goes 5 to 3. The other strand is sort of flipped over, and it goes 5 at the bottom, 3 at the top. So it's sort of upside down. And then they interact in between the two strands. There's an attraction of hydrogen bonds between the two strands, which I'm going to show you in a second. So this strand goes 5 to 3. I'm going to go ahead and put that arrow that I mentioned. This strand goes 5 to 3 with the arrow going the other way. And so that's why we call it anti-parallel, because it's going opposite ways when we're thinking about that 5 to 3 arrangement. So that's called anti-parallel. And then between the two strands, what holds together the two strands are hydrogen bonds. So if there's a guanine on one strand, it will always face a cytosine on the other strand, and they will face each other and they will make hydrogen bonds with each other. Hydrogen bonds are weak individually, but collectively they're strong. And so along the whole double helix, you have lots and lots of these hydrogen bonds forming between the nitrogenous bases on this strand and the nitrogenous bases on this strand. So if you have a guanine here, like I said, on this strand, and let's say the strand is back here. If you have a guanine here, then you have to have a cytosine over here. And if you have an adenine here, then you have to have a thymine here. So these are the rules. DNA will only be built this way. The cell will always build DNA with these matching up these nitrogenous bases. And so every time you have a G on one strand, you have to have a C on the other strand in order for it to hold. If you have an A on this strand, you have to have a T on this other strand in order for it to hold. And so you'll remember what Chargaff's rules were, that A always equals T, percent A equals percent T. That's because every time you have an A, you have to have an equal amount of T because they always bind together. If you have a G, you always have to have a C. So Chargaff's rules ends up being the rules that DNA follows in the double helix, right? Okay, so I've explained about the nucleotides, about the single strand of DNA with the phosphodiester bonds holding one nucleotide to the next, the 5' to 3' directionality of any one strand, and the base pairing rules between the two strands in the double helix. So that is the structure of DNA. In the next lecture segment, we'll then look at, based on this structure, it's actually pretty easy for the cell to make copies of the DNA, but I'll get to that in the next lecture segment.